As Streptococcus pneumonia, let us see about the classification first. They come under the domain bacteria. They phylum is Firmicutes. They comes under the class Bacilli, order Lactobacillus, family Streptococcini, genus Streptococcus, species Streptococcus pneumoniae. General characteristics of the Streptococcus pneumonia. Streptococcus pneumoniae cells are the gram-positive lancet-shaped cocci. Usually they are seen as pairs of cocci, diplococci, but they may also occur singly and in short chains. They are non-spore-forming bacteria, they are non-motile, they are capsulated, and the capsule encloses each pair. Streptococcus pneumoniae take root only in the enriched media. They are aerobes and the facultative anaerobes, the optimum temperature being 37 degrees Celsius and pH is 7.8. The growth is improved by 5 to 10% of the CO2. Identification of the Streptococcus pneumoniae. The microscopy and staining is they perform the gram staining of the sample sputum CSF. The gram staining shows the gram-positive lanceolate-shaped diplococci. Electron microscope image of the Streptococcus pneumoniae. Colony morphology. The colonies on the blood agar plate are small, round, translucent or mucoid with alpha hemolysis, a green discoloration of the agar around the colonies. Alpha hemolytic property and differentiates the S. pneumonia from any species. The alpha hemolytic colonies are further identified by the following confirmatory test. Biochemical identification. The optogen test 6 mm disc with the 5 μg. Inoculate the blood agar plate with the suspected alpha hemolytic isolates. Apply commercially available optogen disc on the streaked blood agar plate. They incubate the plate at 37 degrees Celsius with 5 to 10% of the CO2 for 18 to 24 hours. They observe the zone of growth inhibition around the disc and interpret as the zone size greater than 14 mm indicates a susceptibility, which is a diagnostics of the Streptococcus pneumoniae. Alpha hemolytic colonies with the zone of inhibition between the 9 and 13 mm should be tested for the bile solubility. Bile solubility test. The alpha hemolytic colonies showing the zone of inhibition around the optogen disc between 9 to 13 mm should be tested for the bile solubility. Prepare 1 ml of saline suspension of an organism from the blood agar plate. Bile solubility test. The Streptococcus pneumonia colonies are lysed by the bile. Inoculate 0.5 ml of the suspension into 2 tubes. Add an equal amount of the 0.5 ml of 2% of the sodium deoxycholate in 1 tube marked as test and 0.5 ml of the saline into the second tube marked as control. Shake gently and incubate the tube at 37 degrees Celsius for 2 hours. Virulence factors of the Streptococcus pneumoniae. Capsule. It is the major virulence factors of the Streptococcus pneumoniae, resistance to the phagocytosis. Cell wall. The inflammatory factors. Activation of the alternative complement pathway resulting in the anaphylactic production. During the complement activation, the anaphylactic C3A and the C5A, which enhances the vascular permeability, induce the mast cell degranulation and recruit and activate the polymorphonuclear leukocytes at the inflammation site. Immunococcal proteins include hemolysin, stimulates the production of the inflammatory cytokines like tumor necrosis factor alpha and the interleukin 1b by the human monocytes. Inhibits the beating of cilia on the human respiratory epithelial cell, disturbs the monolayers of the cultured epithelial cells from the upper respiratory tract and from the alveoli. That decreases the bactericidal activity and the migration of the neutrophils. They inhibit the lymphocyte proliferation and the AB synthesis. Pneumococcal surface protein A. It is a surface protein with the structural and the antigenic variability between the different pneumococcal strains required for the full virulence of the pneumococcal inhibition of the complement activation. PSPA enhances the pneumococcal virulence. Autolysin negative mutants have been shown to be less virulent than the wild type pneumococci. Release of pneumococci and the cell wall products. Autolysin triggered by the human lysosome a defense factor released upon the infection and the inflammation. Autolysin thus seems to take the advantage of the protective function of the lysozyme. Complement factor H binding complement, inhibition of the complement activation, inhibition of the phagocytosis, peptide permeases, enhancement of the adhesion. IgE event protease counteracts the mucosal defense mechanism, hydrogen peroxide, and lung injury. Streptococcus pneumonia. The initiation of this pneumonia requires the escape from the mucus defenses and the bacteria migration into the alveolus, firm adherence of the bacteria to the alveolar epithelium and the subsequent replication and the initiation of the host damage. The pore forming the toxin, pneumolysin and the hydrogen peroxide released in the copious amounts by the bacteria disturb the alveolar epithelium and the edema fluid accumulates in the alveolar space. Lipotectic acid, the lipoproteins and the array of proteins associated with the bacterial surface further amplify the interaction with the host. Streptococcus pneumonia bacteria. This bacteria can cause the disease include the pneumonia, ear infection, sinus infection, meningitis, infection of the covering around the brain and the spinal cord. Bacteremia. The bloodstream infection, pneumococcus bacteria are spread. Bloodstream infection, pneumococcus bacteria are spread through the coughing, sneezing and the close contact with the infected person. Symptoms of the pneumococcal disease depend on the part of the body that is infected. They can include the fever, cough, shortness of breath, chest pain, stiff neck, confusion and disorientation, sensitivity to light, joint pain, chills, ear pain, sleeplessness and irritability. In severe cases, the pneumococcal disease can cause the hearing loss, brain damage and death. Treatment. For penicillin, the sensitive strains, penicillin is a drug of choice for the serious cases and amoxicillin for the milder ones. For penicillin-resistant strains, a third-generation cephalosporin is indicated. Vancomycin is to be reserved for the life-threatening illness with highly resistant strains. Thank you.